Hello everyone, this is Lei Shi from Biointron. Today I'm going to talk about accelerating antibody discovery by integrating microfluidics, high-throughput expression, and early developability platform. Many of us here have done antibody discovery, and we know that there are bottlenecks in antibody discovery technologies. The legacy methods can have limited speed and diversity. Hybridoma technology is a workhorse that I personally used a lot, but it can be labor intensive with a low throughput and also have a bias towards immunodominant clones, limiting the diversity. Phage distillery is where I started my career with, and it provides in vitro selection from combinatorial libraries, works on many challenging targets, but it could disrupt the native pairing of heavy and light chains, and often requires extensive downstream and optimization depending on the format of display. So recently, single B cell sorting screening platform provides a trade-off between throughput and formats. And many of these existing technologies can provide a native pairing with using automated functional assays and a higher throughput than conventional methods. But the limitation is also obvious, and many workflows expresses antibodies in single-chain FV fragments requires reformatting, adding time and uncertainty. At Biointron, we developed this platform called AppJob that combines industrial scale, high throughput, direct recovery, and expression of native full-length IgG. So what's AppJob? AppJob is an integrated engine for antibody discovery. It provides scale and can screen 1 to 2 million plasma B cells per run. And also provides speed. And we can screen these millions of plasma B cells in one day, and also obtain paired sequences in 3 weeks, validated. And this this gives us direct access to high affinity mature antibodies from plasma B cells, and greatly accelerate the discovery process. And the workflow, it looks like this. About 1 to 2 million plasma B cells are encapsulated, enriched and encapsulated in picoliter droplets with a fluorescent labeled antigen. And through incubation and FRET detection, we can we can sort these positive droplets into a pool, and then using NGS barcoded NGS technology to obtain natively paired heavy and light chain sequences. And using bioinformatic analysis, we can analyze the diversity and select candidates for expression and validation in about 2 weeks. So we can get a positive sequence in 1 week and get validated in total of 3 to 4 weeks. And this is also supported by our industry-leading antibody production capability with a 2-week delivery. At the Biointron, over more than 100,000 antibodies were made annually. More than close to half of them are bispecifics and multispecifics. We can work on maps up to 3,000 per batch, and from the moment that you give a sequence to purify products for delivery only takes about 2 weeks. We achieve this speed and quality by lots of automation, and from plasmid preparation to dispensing, purification, automated antibody purification using beads-based technology to sample packaging and air quality. This is a case of making 2,700 maps in one batch for our customer, and the vast majority of the antibody was made in in less than 11 days, with a median purity of 99.5% in one-step purification. In addition, we also have extensive experiences in bispecific production. We have to date have experience in more than 260 formats, and with a median purity of 90% for after one-step purification. This high throughput and high efficient mammalian CHOK1-based expression workflow enable us to obtain validated binders from AppJob in 3 to 4 weeks. And next, I will give you a case study looking at PD-1 discovery. We use PD-1 as a validation target often because PD-1 is a well-characterized, clinically validated immune checkpoint receptor. And it also has a dual functionality with the blocking antibodies for cancer immunotherapy, such as pembrolizumab and nivolumab, and emerging agonistic antibodies for autoimmune diseases, such as peresolimab and rosnilimab. It also provides a high bar benchmark for success, and to afford for testing our AppJob's ability with a distinct and opposing biological activities. So, to give a quick summary, we from a single mouse immunized with a human PD-1 antigen, we screened millions of plasma B cells processed and generated close to 7,000 positive hits, meaning positive droplets. And from these close to 7,000 droplets, which we identified close to 3,000 heavy and light paired sequences, and among them, close to almost 500 unique candidates were identified. And the CDR3 length distribution ranges from 2 to 17, with the majority of them in the middle. And using a Catera high-throughput SPR, we analyzed the affinity of a selected set of 52 maps, showing that there's a broad range of high affinity antibodies in this population, and close to half of them showed sub-nanomolar affinity. And many of them bear comparable affinity to pembrolizumab and nivolumab. And there are also 9 antibodies demonstrate higher affinity than the agonist peresolimab. We look at not only protein binding, PD-1 protein binding, but we also look at the binding to PD-1 expressed on HEC293 cells of these 52 antibodies. And 38 of them demonstrate high affinity binding, and 15 of them showed sub-nanomolar potency. Again, using a Catera LSA's high-speed, high-throughput SPR instrument, we did an epitope binning of these antibodies and showed that there can be roughly 5 clusters of binders Cluster 1 resembles pembrolizumab and with the potential blockers. Cluster 2 looks like a nivolumab, and we found only one candidate. And the cluster 3 are much like peresolimab, which is a potential agonist. And there are also 9 candidates belong to novel epitopes. And this provides a roadmap for predicting and validating antibody function down the road. And indeed, we use a luciferase reportage assay to measure the ability of antibodies for the blocking function, and showed that all 17 candidates tested from cluster 1 and 2 demonstrate blocking activity, and some of them are comparable to the positive control, such as pembrolizumab and nivo. Not only that, 2 of them were chosen for testing in the MC38 tumor model in a human PD-1 knock-in mice, and showed that they have a marked inhibition of tumor growth comparable to pembrolizumab positive control. On the agonist side, we use a DCT cell co-culture assay to measure the ability of cluster 3 antibodies to suppress T cell proliferation. And among the for the 10 candidates that tested, they're supposedly to share a epitope that's similar to the agonist peresolimab. The results showed that a robust dose-dependent suppression of T cell proliferation at 10 nanomolar, which is comparable to the benchmark. So the conclusion is a single F-job campaign from one animal successfully identify antibodies with the 2 opposing functions, the potent blockers for immuno-oncology and the functional agonists for autoimmune disease, showcasing this platform's power to deliver diverse therapeutic solutions. Not only that, we also did additional experiment comparing side by side using wild-type C57 mouse to humanized mouse called Hugo. The Hugo mice is a platform from our strategic partner, Cyogen. And the experiment was run in parallel and showed that the Hugo mouse both Hugo mouse and the wild-type mouse generated hundreds of unique sequences. And on the EC50 side, on the protein level, they showed a similar range of PD-L1 binding and also FACS binding on on cells expressing PD-L1 on the surface. And many of them also showed blocking function. And the result between the Hugo humanized mouse and the wild-type mouse are actually very comparable. Beyond that, we have tested different types of antigen, including single-pass transmembrane protein with a highly glycosylated, which is a highly glycosylated complex processing that requires internalization function. And in this case, we identify more than 1,300 unique sequences. And we also tested, we also have experiences in the 6-pass transmembrane protein target that from that particular case, we identified more than 350 unique sequences, and 15 of them actually showed triple cross reactivity between human, mouse, and monkey. On the cell, many many soluble antigens were also used used on our AppJob platform. In this case, in one case, a canine inflammatory cytokine was the target, and we identified more than 300 sequences, and among them, 21 of them were showed a blocking function for the receptor with affinity as high as 200 picomolar. So, in summary, our AppJob platform can in one day screen millions of plasma B cells, and within a week we can get NGS sequence with the paired antibody paired information. In a month, using our high throughput antibody discovery antibody production platform, we can validate these leads in 3 to 4 weeks. So we can identify hundreds of naturally paired antibodies in one time, in a very short period of time. But finding a binder and a blocker is not the end of the story. The path to a successful therapeutic requires a balancing of a complex and often conflicting set of properties. So the early multi-parameter assessment is critical to avoid the later stage failure. This graph showed that on the bottom right, we also need to look at the safety parameters such as off-target binding and non-specific binding. On the left side, in addition to the potency and the affinity, many developability parameters needs to be looked at it from expression level, thermal stability, and the CMC sequence liabilities. And traditionally, this is achieved by using a stepwise screening funnel to narrow down from thousands of leads to hundreds, with a larger scale expression to the final candidates, so in the single digits. But today, discovery process needs to be revolutionized and quickened, and with the reduced timeline requirements and the final preferentially screening in the final format, researchers want to generate data as much as possible and as early as possible to quickly narrow down from thousands to final candidates. And many of these data are also used for AI modeling and AI application. So we think that taking our discovery AppJob platform to a new kind of discovery process that we called rush data. This is our the new Biointron solution in antibody discovery, combining the speed, scale, depth, and integration of the AppJob engine with the high throughput developability capability to deliver de-risked leads. And how do we do this? And we think that we can put together a flexible and custom designed assay package that can be run in parallel or tandem to provide a complete developability profile for thousands of designs per batch. This includes high throughput expression data with antibody characterization and physical stability and purity profile in combination with a non-specific binding test, such as BVP, DNA, and insulin. This is made possible and powered by state-of-the-art high-throughput analytical instruments such as Katera, Sartorius IQ, Octet, and Super DSF, and DynaPro DLS play reader. To combine with automation to ensure rapid and precise parallel analysis of every sample. So when you look back, Biointron can actually start from a high-throughput gene design and synthesis in thousands to high-throughput production of antibodies in thousands with the expression titer purification data to the simultaneous in-depth assay calculation, including binding, thermostability, and heterogeneity and functionality data to provide a vast amount of data to accelerate the project progression. This is particularly useful for fast therapeutic discovery as well as decision-making to support machine learning and AI design. There's a couple of example data that we can provide with a high-speed fax binding SPR using Katera and epitope binding using the Katera instrument, and you can see the same group of epitope families and communities can be grouped in the same color. And using DSF and the DLS, we tested and established a standard reference data set using biosimilars. And for every set of experiment, some of these standards are also included to provide data consistency. And using to uncover the non-specific binding liabilities, we developed assays using baculovirus protein, double-strand DNA, and insulin to establish a standard and consistent, cost-effective assessment of PSR assays. And in each of these assays, 5 controls are included to ensure the reliability of the results. And AC-Sins and HIC HPLC are used often to flag self-interaction, and these are the profiles that we established in-house for the assays. So in summary, from AppJob to Rush Data, the new Biointron solution in antibody discovery is that to combine the high throughput screening of plasma cells to high fidelity sequence library in one day and validate in as little as 3 to 4 weeks, and in combination of the high throughput production called RushMap, and using a custom and flexible design package for assay and data-rich analysis. Combining these 3 key capabilities, we can provide the new Biointron solution for antibody discovery. And the last slide is to give you a general overview of Biointron. We have more than 700 employees and deliver, as I said, more than 100,000 antibodies to our global customers of more than 2,500, including the top 20, almost every one of the top 20 big pharmas. We are based in China, Shanghai, China, and we serve our global customers. And for more information about Biointron, please send us an email or call us and ask about not only the AppJob single B-cell sorting, but also 2-week antibody production, VHH, nanobody discovery, assay development, and ADC conjugation. Thank you very much.